The next speaker is Lihi Adler-Abramovich from the Faculty of Medicine, and she's going to tell us about controlling the physical properties of peptide-based nanostructure by supramolecular polymer co-assembly. Lihi, please.
Thank you. So good morning, everybody. So I would like to thank the organizer for giving me the opportunity to present my work. And so it's really nice to be here, and I really enjoy this meeting, and it's a great opportunity to meet old friend and new friend and establishing new collaborations. So I recently joined the School of Dental Medicine in the Faculty of Medicine, and I'm going to talk about controlling the physical properties by supramolecular polymer co-assembly. Okay, so 2 days ago, I know that Peter convinced you that carbohydrates are very interesting and you should all synthesize them. And I hope that by the end of this talk, I will convince you that proteins and peptides are very interesting. So proteins are mainly the basic structural feature in biological systems. So they are composed of hormones and antibodies and member channels and are in charge of transport and fluid balance. So what are proteins? So proteins are actually a long chain of amino acids. And we are interested in proteins, but mainly in short sequence of amino acids named peptide. And we are in the nano conference of the nano center, so we like simplicity, and we're basically working with the shorter peptide that you can get. You can get a dipeptide, 2 amino acid sequence, and we are also interested in single amino acid. So one of the main processes in nature is self-assembly, and here we have some example of self-assembly of proteins. So we can see the self-assembly of cytoskeleton, the formation of very rigid material like the spider silks, which is also is the results of self-assembly for the formation of other beta sheets. Amyloid fibers, which can be pathological in the case of Alzheimer's disease and can be non-pathological in biofilm of bacteria. And other compounds can go through self-assembly, like the formation of DNA double helix and the capsid formation. So we are utilizing the self-assembly ability of peptides and proteins to form these other structures. So here we can see the self-assembly of dipeptide and the formation of vertically aligned peptide nanotubes, the formation of this other array. And we're also interested in the self-assembly of simple amino acid. In this case, this is phenylalanine, a natural amino acid that can self-assemble to form these amyloid-like structures. And a few years ago, we showed the correlation between the ability of phenylalanine to form all the structures and the pathology of phenylketonuria. So in phenylketonuria patient, high concentration of phenylalanine assimilates in the blood and in the brain of the patients, causing the pathology. But I'm not going to talk about this project today, but rather I'm going to talk about the structurals that we are synthesizing. Okay, so we built a library of peptides, as we can see here. So many peptides that have various amino acids in the sequence, and we modified in either the N cap or the C cap of the peptide. This is diphenylalanine, a simple dipeptide that they're using a lot in our research. And this library can self-assemble into various structures. It can be tubular structures, spherical, plate-like, hydrogels, many architectures. And basically, the sequence of the peptide is responsible to the structures that we will get. It's responsible whether we get spherical structures or tubular structures and what the physical properties will be. But this is not the only reasons for getting a specific structure. So we can see that the environment also affects the structures that we receive by self-assembly. So for example, we have this dipeptide, diphenylalanine, with an N-cap Bock addition, which is commonly using chemical synthesis. And we can see that this peptide can either self-assemble into tubular structures in waters, but the same peptide can self-assemble into these spherical structures in ethanol, and later we show that this is process is this is a kinetic process, and it can be goes from one structures to another and vice versa. So I think that the movie doesn't work here. So I'll move to the next one. So we have this library of peptide that can form various structures, and what is really interesting is their physical properties. So, for example, the peptide nanotubes are stable at the presence of organic solvents, as we can see here, in ethanol and acetone and other organic solvents, and they are very stable at high temperatures, which is quite interesting for bio-inspired materials, for peptide-based materials. They are stable around 200 Celsius degrees. And their mechanical properties are also interesting. So the peptide nanotubes, Young modulus is around 20 gigapascal. It's quite similar to that of cortical bond. And we are talking about peptide that form from self-assembly with the very low covalent binding. Most of them is hydrogen bonding and other bonding. So this is the peptide nanotubes. And how we measure it, we use AFM. We did indentation type experiments. We actually indent the structures, calculated the point stiffness, and then measured the point stiffness and calculated the Young modulus. And then when we work with the spheres that I showed you a few slides ago from the box phenylalanine, we couldn't indent them using metallic tip cantilever. We had to use a diamond tip cantilever to indent the structures, and we measured the point stiffness, and we calculated the Young modulus to be around 300 gigapascal, which is quite amazing for bio-inspired materials. And now we are trying to use these peptide nanotubes and peptide nanospheres inside composite materials that are used in dentistry as fillers to replace the amalgam fillers that used to be common a few years ago. And these fillers has a problem of shrinkage and a problem of limited mechanical properties, and we want to reinforce them using the peptide nanotubes and peptide nanospheres and to form these composite materials that will be very stable and wouldn't shrink upon UV or visible light curing. So I presented a library of many peptide sequence, and we have all kinds of structures, and they have very interesting physical properties. And during the years, us and others show that these nanostructures can be utilized for various applications. So starting with biological application, such as drug delivery and sensitive sensor, through organic light emitting diodes and energy storage devices, and coating of superhydrophobic surfaces, and many more. But we wanted to we wanted to design more features, more new physical properties, and we wanted to extend the diversity of this library. So basically, this library is based on the 20 natural occurring amino acids. We are also using unnatural amino acids, such as DOPA or phenylglycine, and we are using all kinds of peptide modification. But we thought that the next step will be to use co-assembly and to use composite materials. So this is really inspired from polymer chemistry, where co-assembly is very common. And we wanted to apply this on a self-assembled peptide system. Another inspiration comes from biology. So we are working with peptides, and we wanted to mimic a biological system. So for example, we can think of the extracellular matrix, which is a very complex system. It's composed of saccharides, polysaccharides, and protein, and all kinds of components, not only from one building block. So we wanted to make a more complex structures, actually mimicking this kind of complex systems. So I'm gonna show you what we did by co-assembly. So we basically are taking two building blocks with two colors, and we're trying to form new materials composed of these two building blocks and see whether we can control its physical properties. So I'm going to show you 3 examples of projects that we use co-assembly. So one is controlling the peptide nanotube's length. The second is exceptionally strong hydrogels, and the third is controlling of hydrogels' rigidity and durability. So the first project was to try to control the length of the nanotube. So we are working with diphenylalanine, it's self-assembly in solution to form tubular structures. So this is a very simple process, and we get high yield of nanostructures, but the problem is that the length is very varied. So we got a heterogeneous population, and we wanted to control the length of the tubes. So for that case, we used, so this is the diphenylalanine, we use an end cap, the Bock diphenylalanine, the one that's self-assembled into spheres, and we wanted to see whether it can terminate the elongation, the sequential process in the formation of peptide nanotubes. And we were able to control the length. So here we can see just the diphenylalanine, the distribution of the length, up to 30 microns in length. And when we're adding Bock diphenylalanine, here's 20:1, 10:1, or 5:1, we can see that the distribution is narrowing, and the average of the length of the nanotubes is around 5 microns. So in collaboration with the group of Tom and Snolls, we were able to fit it to the Flory model and to explain as termination and breakage of the nanotubes and the formation of homogeneous populations. So we were wondering whether we indeed have co-assembly. Indeed, these tubular structures are composed of both of the components, or do we have just nanotubes of one component and the other in the same environment? So we can see that the kinetics is different. So here is the hybrid, the FF and Bock FF. We can see that the kinetics is different. It's easier to see it here. So this is the diphenylalanine, self-assembly quite immediate, and the hybrid leg face is much longer. And using TOF SIMS, we were able to really image the hybrid. So in green, we can see the diphenylalanine, and in red, we can see the Bock diphenylalanine. And here we can see fibers that are composed of the two colors and basically composed of the two building blocks, which is what we wanted to form. Okay, so this was one example, controlling the length of the nanotubes. And the next one was formation of hydrogels with very unique mechanical properties. So one of the derivatives from our library is the F-MOC diphenylalanine, who self-assembled to form very rigid hydrogels. These gels were used as a scaffold to support the growth of cells and for slow release of drugs. And one new hydrogels that we design and synthesize form these very nice gels, but the problem is that after 7 days, this gel disassemble. So this is an inverted tubes. So the gel is up here, and after 7 days, we can see that it degrades and fall down. And we thought that we might use co-assembly of the F-MOC FF hydrogels, which we know is stable, together with the new amino acids protected moiety that forms the gel, and we'll have a very stable hydrogels. So we started forming these hybrids. So this is component A, this is component B, and this is the hybrids in 3 different ratios. So we can see that the hydrogels are quite homogeneous. We monitor the self-assembly process. So at the beginning, we have this opaque solution, and it becomes clear after a few minutes. We can see that in the 3:1 and 1:3, the kinetics is quite similar, but the ratio of 1:1 assemble quite fast, and after 2 minutes, we have hydrogels, very transparent hydrogel. And what was really nice to see is that the hybrid hydrogels were stable. So this is the only component. This is the image that I showed you in the last slide, the gels that disassemble, and after 20 days, the hybrid is still stable, and actually We then monitor it for 6 months, and we see that this hybrid hydrogels is indeed very stable. So we were very happy to see that it's very stable. And we were interested also in the ultrastructural analysis of this structure. So this is TEM analysis. We can see the fibers composed of these hydrogels, and the one-to-one ratio of fibers are a little shorter than all the other fibers. We did some EDX to show that indeed these fibers are composed of the 2 components. But what was really exciting for us is when we took the hydrogels to the rheometer and we analyzed its mechanical properties. And we see that this is the G prime value of one component, around 3,000. The other is around 10,000. But the hybrid's mechanical properties has a synergistic effect. So basically, usually when we work with 2 components, the mechanical properties are between the properties of part A and part B, something in the middle, so we can fine-tune the mechanical properties. But this is the first time that we got this amazing mechanically strong hydrogel. So we got the hybrid 1-to-1 gave this 200 kilopascal, G-pack volume of 200 kilopascal. And if we compare it to other synthetic or natural other hydrogels, so these are our materials, and here we have collagen and chitosan and other low molecular weight hydrogels. So we quite amazing, strong gel. And later we use fluorescence to show that indeed the 1-to-1 ratio that forms the synergistic effect as a new fluorescence peak, which shows that we have a new materials that assembled from both the component. So what can we do with these materials? And we are coming from dentistry. So we would like to use these very strong hydrogels to treatment for alveolar bone loss. So alveolar bone loss is very common. It's caused by periodontal disease, peri-implantitis, so tooth extraction, and can be caused even by trauma. And now we are developing these hydrogels for tissue engineering and regeneration of bone inside the bone cavity. We would like to use these injectable hydrogels as a scaffold for the formation of new bone. And why it's important that we have this kind of very strong hydrogels? So the group of Ulin from City University of New York, Wayne Ulin from City University of New York, just published recently the use of hydrogels in various rigidity, and they show that rigid hydrogels, because of the mechanosensing of the cells, helps the differentiation of stem cells into bone tissue, as we can see here. So we were able to control the length of the nanotubes, and we form exceptionally strong hydrogels and hope that we can use it in tissue engineering. And the third example that I'm going to show you of co-assembly is controlling of hydrogels' rigidity and durability. So in this case, we are using hyaluronic acid, a polysaccharide, which is commonly used in medicine. So one example is the injection of AHA to the knees for the treatment of osteoarthritis. Another example is the use of AHA in cosmetic treatments for subdermal fillers. And in both cases, the main issue is the rigidity of the hydrogels. So in order to form more rigid hydrogels, cross-linking is used, especially BDDE cross-linking, but the problem is that this cross-linker is toxic, and the FDA approval of its use is less than 2 ppm. So we would like to form rigid hydrogels, and of course we choose the co-assembly approach to reinforce this polysaccharide hydrogels. So the problem is limited durability, and we choose to work with the FMOC-FF, low molecular weight hydrogelatas, and to form these composite materials. So here we can see the composite materials that we form. So the HA looks very similar to water. It's very it's a non-rigid hydrogels, and the addition of FMOC-FF form this rigid three-dimensional structure. One issue in this case for the treatment for subdermal and the treatment in orthopedics is that the hydrogels will be injectable, so we checked that it can go through 40 gauge, a very thin needle. These are inverted tubes of the various hybrids that we are forming. We can see that we have homogeneous solid hydrogel. And what was interesting is, of course, the rheological analysis. So here we have a system that we can fine-tune the mechanical properties of these hydrogels. So we can start, so this is a commercially available compound. This is cross-linked HA named stillage, which is used for subdermal application. And using the addition of the low molecular weight hydrogelator, we can elevate the mechanical properties and we can fine-tune. If it's a subdermal fillers, we don't want it to be too rigid and our face not to be able to express itself. So we want to control, and this is a nice way to fine-tune the mechanical properties without any cross-linking, without any toxic cross-linking, with a stable low molecular weight hydrogel. So later we showed that these peptides are safe, its cell viability, and we did some toxicity assays in mice, and some preliminary results show that the hydrogels is stable after 30 days injecting subdermal to mice, and further study should be done on its durability and degradation process. So to summarize, we designed various peptides. We have this library of peptides that can self-assemble into tubular, spherical, hydrogel, platelet on color ultrastructures. To extend the diversity of the nanomaterials, we use co-assembly, and I hope I convince you that co-assembly is a very interesting approach for diversity. So we were able to control the length of the nanotubes. We formed extremely rigid hydrogels with G prime value of 119 kilopascal. We formed stable hydrogels for injectability in orthopedics and cosmetics. And before I finish, so some more activities that we have in the group. So we are interested in biofilm. This is very relevant to the dentistry. So we're trying to form this model of biofilm and to form antibacterial dental composites. We are coating dental appliances to make them less to make less biofilm attached to it, to make it more hydrophobic and to change its properties. So we are also using microgravity as a model system for screening of drugs against osteoporosis. And we are using collagen and mimicking of collagen by short peptides to form scaffold for tissue engineering. And I would like to thank my group. This is my group in the dental school, and to thank my collaborators and the foundings. And we are looking for talented students, PhD and MSc. So if someone is interested or knows on somebody is interested, so please contact me. And I would like to thank you for listening.
Thank you, Lihi. The paper is open for questions. Please, Amit.
Do you know what is the chemical nature of the better behavior or the strength of the system that you get? Or can you use it to develop new types of
So basically, most of the molecules that we are using, if we are talking about the mechanical properties and the G prime values, so it's derived from the peptide sequence, and we cannot control. We can only fine-tune by mixing two components. And the only case was the one that I show here, and we think that we have new hydrogen bonds, and actually now we are trying to crystallize this co-assembly and to understand the chemistry behind. Yeah, of course, it's very interesting. So we have crystals, so I hope we can solve them, and then we'll know a little bit more about the chemistry inside, but it was actually quite surprising to get these amazing parameters.
My question, is it obvious that cross-linking is not a possibility in these systems, that you would like to introduce it to increase rigidity or
Yeah,
or
so
strength?
cross-linking, so I mentioned it with the HA, so cross-linking is the commonly used product now. They are cross-linked, but the problem with the cross-linking is that it is toxic. So it's a safety
No,
and
I mean in your system. that you
if we have cross-linking?
Three-armed amino acids that will cross-link to other self-assembly fibers.
So we don't see new covalent bonds in this system specifically, because in other systems, when we heat it, when we get some energy into the system, we get cross-linking, we get new covalent bonds. But here we see only a shift in the beta sheet or alpha helix structures, and we can see we have hydrogens bond, we have van der Waals bonds, but we are not having any covalently new bonds. But this is something I hope to get from the crystals that we are trying to have.
Okay, thank you. Other question, please?
You show the tube you're getting, I think was it the second subject, you managed to reduce their monoviscosity towards 5 nanometers. I think, or was it micro?
So it's the first project about the short peptides, we can confront the length.
Yeah,
Yeah.
we've caught something.
Yes.
So the sizes were reduced to 5 nanometers or
No,
microns?
5 microns. This
Microns.
is the length of, so we're talking about nanotubes because of the diameter, so they are approximately around 100 nanometers in diameter, and the length is between, so we started with very dispersed, it can be few hundreds of nanometers until 25 microns, and then we can narrow it down to an average of 5 microns.
Okay. Did you try taking this vanilla and co-assemble it covalently with the polymer? Still self-assemble into a structure. If we attach it, let's say, with an egg or a forearm egg.
Yeah. So basically the peptide that we are working on. So basically the peptide that we are working are very small. So if we attach something which is too big, it will affect the self-assembly. We do have some experience with short moieties of PEG, and we have a similar self-assembly. So the diphenylalanine stacking, this is a pi stacking procedure, that derives the self-assembly, indeed assemble into other structures with a quite similar morphology. But if we are attaching too big of a molecule, so it changed the solubility and changed the resulting structures.
Any other questions? If not, let's thank Lihi
Thank you.
again. Thank you. The last